And I'm pleased to introduce Dr. Nikhil Hirani. Nick is a consultant respiratory physician at the Royal Infirmary in Edinburgh and principal investigator at the Centre for Inflammation Research. In addition, Nick is a member of our medical and scientific advisory panel, so we are very, very grateful for the time he gives to us. Nick, over to you.
Thanks, Graham, and thanks to Sarcoidosis UK for inviting me to talk. I'm a chest physician, and so my talk is really about chest disease, although you've heard other speakers this morning and Anne-Marie talking about other aspects of sarcoidosis. I've dipped into the morning session and to Anne-Marie's talk, so really high quality talks. So the first thing to say is just lower your expectations, relax. This is a wander through around 30 minutes of clinical trials, mostly focused on lung disease. Next slide, please. Great. I have no declarations of interest, although I am PI or CI to some of the studies that that I'm going to talk to you about. So there's 5 sections of the talk. What are we treating? Why is it so difficult? What progress have we made? Is the future bright? There's a spoiler alert, yes. And what can we do to help? And by we, I mean everybody in this forum, in the wider community. So move on to the next slide, thanks. So you've been familiar with this kind of diagram before. Sarcoidosis affects all parts of the body, perhaps except the adrenals, but the lungs are affected in most patients. And so as I'm a lung doctor, I'm going to focus on that part, although some of the trials that I'm speaking about have entertained or have been the subject of other organs. So continue to the next slide, thanks. So what is the enemy? What is the thing that we're dealing with, which is the granuloma? So you've heard of that. That's the nasty beast that's in the top left panel, and this is a disease that manifests by developing granulomas, which is a structure of cells. Oh, sorry, too far ahead. Thank you. Structure of cells, mainly macrophages, lymphocytes, fibroblasts, other cells that form a unit. We don't know what the instigator of the unit is, but that granuloma is the thing that we're trying to address. And the bottom panels are a CT scan of the lungs and a corresponding PET scan. A PET scan is a scan that shows activity of sarcoidosis in this case. And you can see the PET scan, which is the kind of red, orange pattern in the lungs. It looks like a fire. And that's what's happening in active sarcoidosis. It's an inflammatory fire, in this case, in the lungs, but it's the same thing with other organs. And the aims of dealing with a granuloma or this fire is stop the start of the fire, stop the spread of the fire, and stop any lasting damage. And the lasting damage actually is due to persistent inflammation that's causing fibrosis or end organ damage. So that's the 3 aims, and that's the enemy that we're dealing with. Move on to the next slide, thanks. So we have a friend, or is it? So we have a good treatment for sarcoidosis and putting out fires, which is steroids. And we all know about steroids. And I say it's a friend because it's a very effective treatment for most people with sarcoidosis for any organ, but it's obviously a foe as well. And most of the audience will, in fact, all of the audience, I think here, will know the common side effects of corticosteroids. So although it's a very effective treatment, at moderate or high doses, or for a long time, or even at small doses for a long time, it causes sometimes intolerable side effects, which I've described here. And most people have taken steroids. If you've ever taken steroids, you'll be familiar with that. So clearly there is an unmet need for newer treatments in place of steroids. Although the steroids themselves, sometimes we don't know the actual dose or the time that we need to treat the patients, even for a treatment that we've had for 50 years. And I'll mention some of that briefly later. So next slide, please. So why is it hard? Yeah, so this is the brick wall of sarcoidosis. And this is some of the bricks, the obstacles that we face. And I hope you can read that. They'll be very familiar to you. We have steroids already, so why are we bothering with new drugs? Well, I've already described, and everybody in the audience knows why we need some other drugs rather than steroids. Sarcoidosis is kind of it sits in the middle of, it's not common enough for everybody to know about, but it's not the ultra rare disease that really attracts attention, that gets onto, Radio 4 in the morning. It's, it's that kind of state where it's in the middle ground, and it's really, that's a problem for sarcoidosis. There's no clear pathway to drug approval. There's no regulatory pathway approval because we haven't tested a pathway to regulatory drug approval because we haven't had new drugs for a long time. Clinical trials are too hard to enroll to. So, that's a common problem for lots of clinical trials, but sarcoidosis is really susceptible to being difficult to recruit to. We don't understand the mechanism of disease. Well, that's true. We don't understand the mechanism of a lot of diseases, but that doesn't mean we don't have treatments for a lot of diseases. Sarcoidosis impacts underrepresented groups. Well, that's absolutely true. We've heard before in the morning, I think, that underrepresented group, ethnic minorities, socioeconomically deprived people who get sarcoidosis more commonly than other people, for example, don't really engage in healthcare sometimes and don't engage with clinical trials. So that's a problem. Sarcoidosis is complex and varied. Well, we've heard a lot of information around this morning that is true. It is complex and it is varied, and there's a lot of organs that are involved. So getting a unified therapy for sarcoidosis is going to be challenging. And then we don't know how to measure the treatment response for a clinical trial, and that is true. And again, we've heard some of the symptoms of sarcoidosis that are very varied, including fatigue, but sometimes it's difficult to measure fatigue, and it's very hard to conduct a clinical trial, for example, that measures fatigue, for example. So there are lots of things, lots of challenges. This is the brick wall of sarcoidosis that we need to battle. Next slide, please. So last year, I didn't go to the UK sarcoidosis talks, but I watched some of them, and Katie Beckman gave a really nice talk where she gave the sort of history of sarcoidosis treatments. And I encourage you to go to Katie's talk and the previous year's talks because they're very good. And I stole, actually borrowed this slide from Katie. She gave it to me. So this is a slide for the historical timeline for drug treatments in sarcoidosis, starting, and the traffic lights mean that these trials, green lights, positive trials, yellow is need more information, and red means that trial is effectively negative. So I'm not going to go through all of that because that goes up to around 2023, 2024. Next slide, please. But I'm just going to focus on maybe 5 studies that are most recent in the last 18 months or so. Some of these studies are completed, some of these studies are still ongoing, but I know that the audience that's listening today is going to be interested in up-to-date clinical trials. So I'll go through those one at a time, and the format will be the same for all 5 trials. So next slide, please. Great. So this is a trial that it's called the Sarcoid trial, and it's published in 2024. And I'm bringing this up because it's not a new drug; it's actually steroids. And I was hinting that we use steroids all the time, but it's despite 60 years of, almost more than that, 60 years of steroid use in pulmonary sarcoidosis, we actually don't know what dose to use in terms of a clinical trial. It's not been proven what the most effective dose to use, and it's not been proven how long to treat people for sarcoidosis. Every clinician knows approximately what dose to use and how long to treat for, but it's not been ever proven in a clinical trial. And so this is, I mean, it's taken many, many years to come to this point, but there is a clinical trial published last year. And I'll just talk you through it. So this asks the question, is high dose, which is 40 milligrams in this case, 8 times 5 milligram tablets, versus low dose, 20 milligrams of prednisolone, is that effective in treating lung sarcoidosis? Which is more effective? So this is 86 patients recruited. All of the patients had lung inflammation, a fire in the lungs, not necessarily detected by PET CT scanning, but all of them had pulmonary inflammation due to sarcoidosis. And they randomized the patients into having low dose steroids, 20 milligrams, versus high dose steroids, 40 milligrams. And then they tailored the dose down over 6 months to a maintenance dose, and then they stopped it. And then they followed up the patients for at least 18 months and asked the question, how many patients relapsed with the sarcoidosis? And the bottom line is that 20 milligrams versus 40 milligrams is no difference in terms of relapse rate. About 40% of the patients relapsed and had to have more steroids at the end of the 18-month follow-up. So that's quite important. In other words, 20 milligrams in this study is as effective as 40 milligrams. But there's other things in the study that are important to describe. One is that they actually did some quality of life questionnaires as well, and they measured some quality of life in their patients, and actually hoping that 20 milligrams would be better tolerated than 40 milligrams. Effectively, the side effects were similar in both groups, in the 20 milligrams versus 40 milligrams. But if 20 milligrams is as effective as 40 milligrams, there's no reason to not use 20 milligrams in this study. And the other thing is that the relapse rate is quite high, 40% of patients relapse. And one of the reasons might be that the treatment period is only 6 months. Now, most clinicians, especially in the UK, I think, would advise patients to be treated for longer than 6 months. For pulmonary sarcoidosis, at least 12 months. 18 months, I think, is reasonably standard. So 6 months is a relatively short time. So that this study isn't ideal because most physicians would use a longer period of treatment, and therefore the relapse rate might be less than 40% if you treat for longer, but that's unproven. The other thing is that this is not a blinded study. Patients knew they were on 20 milligrams and 40 milligrams, and the physicians who cared for the patients knew that they were on different doses. So it's not a blinded study, and that's really important. As we go on to seeing the next few studies are randomized controlled blinded studies, which is the gold standard for a clinical trial. But this is an interesting study because I point it out because even steroids, which is the standard treatment, there's a lot of stuff we don't know about the dose and the time of follow-up. Okay, next slide. So this is the first of the 4 newer studies. So this is Resolve Lung. a phase 2, which is so clinical trials go from early phase, which is phase 0, phase 1, through to phase 4, which is after the drug has been approved. But phase 2 is like the middle ground of a clinical trial. It's a randomized double-blind placebo-controlled trial of something called a CSF antibody. And that antibody targets GM-CSF, and GM-CSF is one of the molecules that we think is important in the development and the persistence and the spread of granulomas. So interesting study, 107 subjects, which is quite a large number for patients with for a sarcoidosis study. All the patients had fire in the lungs, active lung disease, and all the patients had background therapy already on steroids, and sometimes already on other medication as well as steroids. And they were randomized to either an injection, serial injections of this drug, or a placebo injection over time, 28 weeks. And the endpoint was how many patients relapsed or had an escalating use of steroids during the treatment period. And effectively, disappointingly, the study has been negative. It's not been published yet, but there's been articles on the website suggesting that the data is negative. As many patients relapsed with the study drug compared to the placebo arm. So it's disappointing, but it's good to some extent in that it's feasible. It shows that 107 patients can be recruited to this type of study with serial injections. And therefore, we shouldn't lose hope of this study, although it's disappointing that the study hasn't shown a benefit. And I suspect this drug isn't going to be taken forward for future clinical trials. Next slide, please. So the next study that I'm going to talk about is a larger study. Now, this is really exciting. EZOFIT, a phase 3 randomized double-blind placebo-controlled trial showing the efficacy of something called efzofitimid. I've been practicing how to say that, and I've mangled it, so I'm pretty sure that somebody will teach me. A drug that targets neuropilin-2. Again, a molecule that's said to be important in the formation and perpetuation of granulomas. Now, the most exciting thing, because the data hasn't been published yet, the trial has been completed. The most exciting thing about this is the investigators, to their credit, have recruited 268 subjects, by far the largest sarcoidosis clinical trial globally that there's ever been. We're excited to see the data. We don't know the data, but it's a year-long study, and it's an infusion of drug, placebo versus the drug arm. The patients were on steroids at baseline, perhaps on other drugs as well, but they have to be on steroids even at low dose. We don't know the data, the outcome of the data. We will know it maybe in a few months' time. But the bottom line is that it's a success anyway, because it shows that we can recruit that number of patients. We can do a phase 3 clinical trial in sarcoidosis. It's the first time we've ever been convinced that we can do it. And I think that's a success story in itself. Hopefully, fingers crossed, the data will be positive, which will be the first effective new drug for a long time, but wait and see. The next slide, please. And then this is the XTMAP study, which is an earlier phase study, phase 1 and 2. Again, we don't have, this is an ongoing study, we don't have the data. We know that the first cohort has been completed, 39 subjects, and this trial is going on in the UK as well. It's on UK Sarcoidosis website. The first cohort has been completed, which is a dose-finding cohort. In other words, the investigators needed to know what dose was optimal to use in the later cohorts. The patients all had pulmonary sarcoidosis, at least pulmonary sarcoidosis, active disease. They all had steroids and sometimes other drugs as well. They were randomized to a TNF antibody or a different type of TNF antibody than we normally use in clinical practice. It's a 12-week study between the TNF antibody and a placebo. The data isn't known yet, but the promising thing is it seems safe. We now know, or the investigators now know, which dose to use. Next slide, please. And we are going on to recruiting 62 further subjects, a longer study now with 24 weeks, with a known dose that's been tested in the earlier cohort and versus a placebo, and that study is still recruiting. So again, it's promising. Again, we are showing that we can recruit patients who have sarcoidosis that are resistant to treatment, and hopefully this data will be promising enough to be to suggest that we have to do a phase 2b or larger studies. Great. Next slide, please. And then the KITE study, which is again study that is ongoing. It's ongoing in the UK as well. Around 100 subjects will be recruited. I'm not sure how many patients have been recruited, but the numbers are reasonably good. Again, patients have ongoing lung inflammation. Now, this is an interesting study because in the previous studies, all the patients had to have already had been treated on with lung inflammation with steroids or other drugs or other drugs. So this study is different because these patients have lung inflammation, active lung sarcoidosis, but are not on steroids. And I know that patients, when I talk to patients about starting treatment on steroids, particularly if the patient has very few symptoms or manageable symptoms, and the lung function isn't particularly poor, and the symptoms are manageable, then sometimes the patient and the physician thinks, well, actually, there's a gray area here. Do we need to treat with steroids? Because it's a long journey with steroids, and steroids have side effects. Do we need to treat with steroids to prevent ongoing damage? Or can we wait and see and monitor a little bit further and see if the sarcoidosis develops into more aggressive sarcoidosis? So that window of opportunity is where this patient study comes in, because that window of opportunity is ideal for patients for this study, where this drug is called a chitinase inhibitor, and chitinase is the protein that's expressed in cells that form granulomas, the macrophages. And this study directs the drug to chitinase and hopefully prevents granuloma formation and persistence. So, and it's good for patients that are ambivalent for taking steroids, and where the physician thinks the steroids aren't urgently required. It's a 12-week study, randomized to the drug or placebo. It's still recruiting, and the endpoint is how many patients will have to require steroids, but particularly how many patients have ongoing inflammation on their PET-CT scan after the 12 weeks of treatment. So the patients undergo a PET-CT scan at the beginning of treatment and a PET-CT scan at the end of treatment. So it's a really interesting study, and I hope that if you haven't known about the study, then the details are on the UK Sarcoidosis website. Okay, next slide, please. Thanks. So why does it take so long for the new drug developments in sarcoidosis? So Can you just hit the next slide? Thanks. So it takes long because of the top panel. This is the pathway to drug development, starting with the target selection. In other words, what is it we're trying to treat? And then lead discovery, medicinal chemistry, in vitro, which is cell-based laboratory work, and then in vitro, which is usually animal-based work. And that takes a lot of time. And in fact, that top panel takes years and years to develop into a potential drug, And then, next slide, please. That's even before we get into the patients. And the next phase, actually a patient studies phase, early phase to later phase studies, takes equally long until we get something called a regulatory approval, which is a successful phase 3 study. Now, I think that journey from, discovering or thinking about what target to address all the way to a phase 3, the end of phase 3 study, is typically between 10 and 20 years. It's a long time. The top panel is sometimes between 5 and 10 years at least, and the bottom panel is equally long. So, and that's because it's difficult. So is there any way we can speed that up? Well, possibly yes. Next slide, please. So is the future bright? Yes, it is. Next slide, please. So one of the things that we said from the start is, the target selection. How do we choose which target is best? So I've given you 3 or 4 examples of clinical trials that have targeted certain molecules. How have they come about by these molecules? How have they discovered these molecules, and how have they developed a drug that can go through to a clinical trial? Well, that's called target selection. Now, the problem is that the granuloma, as you imagine, is complex. There's a lot of cells, there's a lot of mediators, and that graphic in the middle graphic is around the granuloma is highlighting just a handful of the potential molecules that go up to that putatively or plausibly go into making up a granuloma. There are hundreds and hundreds. So how do we rationalize hundreds and hundreds of potential targets and then go on to which is the most plausible target, and then go on to making a drug that's targeted to the target? It's very, very difficult. And if we do it manually, as we have done it in the past, it will take years and years. And that's how, and that's why we use AI or generative AI. So we've hinted, and Anne-Marie's hinted that at the data. So if I gave this talk 5 years ago, I wouldn't have mentioned AI at all, really, even though researchers and big pharma were using AI even at that time. Even if I gave the talk a year ago, I would have mentioned AI, but I would have thought, well, it probably won't be a game changer right now. It might be a game changer in a few years' time, but clearly it's a game changer right now. And even right now, it has sped up the development of potential drugs. And next slide, please. So give you an example. So I don't want you to read this graphic on the right-hand side. This is one of several studies that I could have cited that have used AI to generate a molecule, a potential medicine that went into, that could plausibly be used in patients. So the graphic on the right-hand side within the loop is effectively the same graphic as I've shown with the hexagons in the left-hand side, which is the drug development pathway. And these researchers were not looking at sarcoidosis; they were looking at idiopathic pulmonary fibrosis, which is another interest of mine. So they asked the question, can we generate, using AI, plausible targets for idiopathic pulmonary fibrosis, just using some knowledge of what we already know? Can we choose the plausible targets? Can we choose the most druggable targets, the targets that have least likely to have side effects, the targets that are most likely to be targets for medicines because they've already been tested for repurposed drugs, for example? And they went through this pathway, and they arrived at a short list of drug targets. They designed a drug, all in silico, all with computational biology rather than experimentation. There are some experiments, obviously, small experiments in this study as well, but mostly this data is driven by computational biology. And they designed a drug that they used in healthy volunteers at microdoses, which is called a phase 0 study or a phase 0.1 study. The beauty of this is they did all of that from the first inception to the patient study, or the first in human study, in 18 months. Now that's remarkable. That would have taken a decade ago 10 years, 5 to 10 years. Even 5 years ago, it would have taken a long time. And this is just one example. We now know that there are about 30, 40 drugs that are in very early phase studies that have all been designed basically through AI in silico computational biology, and all within a very short time frame. So the first part of this drug discovery pipeline is being sped up significantly through AI. Of course, the algorithm, the AI, is only as good as the some knowledge that the large language models can soup up, and it's only as good as the algorithm itself. But clearly there is huge potential in this, and it is the future. Next slide, please. So I hinted that that AI can speed up all of this first stage of drug development, but even with AI, there will be hundreds of plausible targets, hundreds of potential medicines that will come about. If we are thinking about testing each one of those medicines through early phase clinical trials in a disease area such as sarcoidosis, where traditionally it's not common and it's difficult to recruit to, we will still be years and years in the making of a new drug. It's not going to be quick if we do it in the old-fashioned way. So can we speed up this next stage of testing lots of drugs quickly? Well, that's hard, and I don't believe there's an easy solution to that. But one solution potentially is some work that we've been doing in Edinburgh, which is phase 0 lung microdosing and sampling. So this is the left-hand panel is my colleague Dr. Akram, who is bronchoscoping a patient, putting a telescope down into a patient who has lung inflammation. And push on to the next slide, please. And what he's doing is the bronchoscope is being put into in this case, into the left lung. And he's taking some samples from the left lung. That the samples will contain cells and brushings of cells and fluid. And then he's instilling a very small dose of a drug. This is a drug, it's a novel drug that's never been in patients or even human volunteers before, but it's a very small dose. And he's going to do the same in the right lung, taking some samples and instilling a placebo. In other words, not the drug, but the vehicle of the drug, a placebo. And then he's going back to the left lung and he's sampling the same cells, the same area of lung, to see if the fluid and the samples in the cells from the fluid show a change after the drug delivery. And he's going to compare that to the other side, the placebo-controlled side. So this basically is an experiment in humans that we would normally do in a test tube. It's taking human samples, instilling a drug, and testing if the drug targets the actual molecule that is meant to target and changes the behavior of the cells. And on the right-hand side is the orange dye, the blue dye shows you where the drug is being delivered, which is a small area of the lung. And the beauty of that is that it's a very safe way of delivering a drug that's not known, not been tested in patients before, because it's a very, very tiny dose. It doesn't have clinical efficacy. There's no way that we can cure sarcoidosis or any disease with this type of treatment. It's just a way of describing or defining if the drug actually acts on the cells in the human body in the active disease. So we've just completed the first study of lung microdosing with a new drug in patients with lung inflammation and fibrosis. And that scenario is one which we need to explore further. It's possible to deliver more than one drug at one time, and that means that you can speed up the process. But if you can deliver more than one drug at one time, then you can describe which drug is most effective in the human lung. Okay, next slide, please. Next slide, please. So I've hopefully kind of convinced you that that we have sped up at least the first part of this program, and maybe even speeding up the very early phase human trials. But as you probably know, if you know the fable, the hare doesn't actually beat the tortoise in the end. And so clearly, AI is not going to be the answer in all circumstances. The hare can get us so far, but in order to get a new drug into regulatory approval, it can't all be done with AI or even any other novel technology. Next slide, please. So that brings us to what can we do to help. So this is Colonel John Boyd. He's a military strategist, worked in the Pentagon for many years. He was an Air Force pilot. And this quote, you might have heard this quote before. People, ideas, machines, in that order. It's to remind us, to remind everybody, that actually we can't do anything just with machines. It's people and ideas that drive us forward. And in order to overcome some of these obstacles that we've described before, it's not going to be machines and computers. It's going to be us as a community, including everybody in the core, building a community in order to break down this wall. Next slide, please. So some of these obstacles, these bricks, some of these will be improved with novel technology, but some of it will only be improved with increased education, lobbying, and engagement from all of us, effectively. Incidentally, if you're wondering how I generated this picture, I used AI. So obviously, I asked one of these large language models to generate a photorealistic picture of some people with some tools that look as if they are going to break down a wall. So, and this image was generated for me. Next slide, please. So I thought, well, actually, that's pretty good. I couldn't have made this on my own, so that's good. So I said to the large language model I used, oh, this is good. But Anne-Marie spoke about bias, and I have bias, and you have bias. Everybody has bias. And I looked at these pictures and I thought, well, actually, I'm talking to a UK audience, and my personal bias is that I think these people look American. Now, I don't know why they look American to me, because I know that I'm biased, but I suggested, well, they look American, and maybe could you make the people look more British? I just wanted to test if what the AI model would do, because I have bias, and didn't know if the AI model would have bias as well. So next slide. This is what the AI model generated. Slightly disappointing, really. It just looks a bit washed out. All the people just look slightly older than the previous photograph. So that's really a warning or an illustration that AI, these kind of tools, are as biased as we are, because they only learn from us. And it's hopefully it's an amusing illustration, but it's a warning as well that everything we use technology based is driven by what the algorithm feeds in, what the algorithm feeds on, and that is based on human data. So we need to be very wary of that when we use these large language models in terms of drug development and onwards. Okay, next slide, and I think the final slide. Thank you for listening, and thanks to Sarcoidosis UK.
Thank you very much, and congrats on getting all those drug names right. So,
oh, I don't think I did.
There must be an AI model to actually get them translated for you, I'm sure. But yeah, thank you. We have time for one question, which I think is a really important one. So I'm going to throw that at you now. We often see adverts for medical research. What should we be considering, though, before signing up for one, and how risky actually are they?
Yeah, so the quick answer is that they are low risk. So so there are phases of trials from early phase to late phase. The later phase trials have had more patients already recruited into them. So in theory, they are safer, because the more patients that have already been recruited, the safety profile of the drug is better understood. But even with early phase trials, they have been through rigorous approvals before. They've been tested in vitro and in animals. And it's really important, because we can't get late phase trials without early phase trials. And if you enter into an early phase trial, you will be rigorously monitored for safety effectively. So no trial is without risk, but in fact, no medicine is without risk, of course. So I think I would encourage people to read the literature. The patient literature is very, very patient focused. So it's designed to tell the patients the risks and benefits of entering into a clinical trial. And I'm so impressed by the altruism of our patients. They commit to clinical trials where they don't see any direct benefit to themselves, but they know there's a benefit to the wider community.